Today it's Jesper presenting his licentiate thesis. We are two supervisors here. It's Claes Lundström from Center CMIE here, CMIE, and it's me, Morten, from Applied IT, Interaction Science Honors, GU. The discussion leader of today is Professor Ivan Yoda from TU Wien, and we're very happy to have you here this precious time. And the examiner for Jesper's PhD education is Professor Devdat Dubashi. He could unfortunately not come today. We've tried our best to fit things together. However, we will provide him with all written and video material after this session so that he will get all material he deserves to have as examiner of the process. So the title of Jesper's work is Designing a Digital Pathology Workstation for Routine Practice. So welcome, Jesper. Give me the word.
Yes, I'll stand somewhere here. It was very heavy on this side of the room for some reason. Morten is a little bit scary, I guess. Okay, so I will talk for around 40 minutes to present the thesis. For those of you who are not familiar with pathology, I will start introducing what pathology is so that I can explain what digital pathology is. And then I will go through the papers in order. If you haven't grabbed a copy and you want one, please feel free. And then I'll end with a discussion to start off the discussion with Ivan. And a special thanks, of course, to Ivan, who come all the way from Austria to be the opponent here. So does this thing work? Yeah? Okay, so what is pathology? A simple definition is the study of disease under a microscope. Welcome. And most larger hospitals in Sweden and most Western countries have a separate pathology department, which all the clinicians can send small pieces of specimens for analysis to. It could be, for example, when you take a biopsy with a needle, you insert a core needle and you pull out the small core and you put it in a plastic bag and you send it off to the pathology lab. It can also be if you have a suspected skin part, you cut it off, put it in a plastic box and you send it to the pathology lab. Or larger operation specimens where you have an operation, you cut up a tumor, you put it in a plastic box and you send it to the pathology lab. And they end up here. For larger specimens in large plastic boxes, you can't really look at everything. So you have to pick and choose a few representative pieces in order to do the further analysis. So typically a technician selects pieces for further analysis and put them in these small plastic containers. Which is then put in a big machine where it's dehydrated, which is a process you do in order to stop the naturally occurring deterioration process. After this machine, you embed it in paraffin in order to create these stiff blocks, which allow to use the next machine, which is a cutting machine, which cuts thin slices around 5 microns thick. So you cut with this machine and they end up in this water bath below. And then you pick a glass slide, and at this stage, the tissue is almost transparent. So if you put it under the microscope, you don't see anything. So as the last step, you add color. And this is how they used to do it, and they still do for rare stains. But for the most common ones, you put it in a machine and you select the program. And they become out this nice and pink. And most of the steps before, all of them can be performed with a machine, except the cutting, which really you need fine precision work that only humans can do today. These are then stacked in piles and sent off to the expedition of the pathologist, where they go through the glass slides from each case in order to analyze them. this is how it looks like. This is a quite typical microscope, I would say, especially in a university hospital where you have teaching, because it has a second eyepiece where you can cooperate with people, you can get help, you can look at cases together, and you can teach your students on how to do this work. So the pathologists get the cases, look through the slides, switch the lenses in order to zoom around, and move around the glass slides in order to look at different tissue structures. Dictate the report, typically, which is then sent back from wherever the plastic box first came from. So this is what the pathology department do. And it's a sort of a service organization. So you as a patient, you never go there yourself and you never meet them, but other clinicians use this to get help with harder diagnostic work. Most of the work is cancer diagnostics, which makes up the full workload, but there are also other types of analysis that needs to be performed. So what is digital pathology then? It is, you do all the steps the same, but in the end, instead of analyzing with a microscope, you put them into, welcome. You put them into one of these scanners, which is basically a moving microscope on a stage that moves around all the slides and create digital images of the glass slides. And because it has around, the typical scanning magnification is 200 times, it creates images which is typically 50,000 times 20,000 pixels large. So around 1 gigapixel and up. If you can also scan in 400 times and then they become larger, of course. A good example to get a feeling of the size of the images is, I think, this comparison. You have Stockholm to the left and you have a breast cancer to the right. And I have now matched the magnification so that a typical cell is as the same size as the typical building in Stockholm. And I also take the opportunity, of course, to compare Stockholm with cancer whenever I can. And so you can sort of imagine the work of the pathologist as zooming around in Google Maps in Stockholm and analyzing the growth structures of the buildings. And before we go further, I think it's just worth stop here and have a quick look on a typical image. So again, this is a, I can do it like this also. And this is a typically invasive breast cancer, and where you can, in a digital image, you can zoom in all the way around, where you can see these are nuclei of cancer cells. The pink stuff here is the response of the system, bindväv in Swedish, the stroma in English. And these large white reservoirs are fat cells, which is, in a breast, is the most common cell. So we'll look at more of these images later, but just now I want you to see what it is that I'm talking about. Oops. There, right? Okay, so the reason I'm probably here and why we work with digital pathology in Sweden is probably because of this pathologist in Kalmar, which his name is Sten Torstensson. And in 2006, one of his colleagues got a problem with his cervical spine, because when you sit in front of a microscope, you really have to sit still with your neck when you're working. And when you do that 8 hours a day, every day, it's quite common to get some sort of neck and neck problems. So they tried to find a solution for it, because he couldn't really work full time anymore. He couldn't stand working that much in front of the microscope anymore. So they were looking for solutions. But then this new technology came around, which made it possible to actually digitize all the images. It was, I think, the first scanner that could be used for this purpose came out in 2005. So it was really new technology then. So they tried the technology out and try if it worked out to diagnose it digitally, and it worked well. And this is what, when I started on this project 2.5 years ago, the first paper I wrote together with Sten in order to try to summarize all the work that has been done. And it became the first paper in this thesis. My contribution to that paper was more as an outsider, try to understand what the peculiarities of that work was. And we also did a questionnaire which were sent out to the pathologists in Kalmar and Linköping in order to get a quick feeling of how this type of system worked and what was good with it and what was some simple problems. First question was this one: how much do you use it? And we asked them, like, how many of the cases that pass through your office do you choose to diagnose digitally instead of with a microscope? And you can see a few enthusiasts using it a lot, whereas the big bunch of people just use it a little. And he also asked what they used it for, what type of features in the software they use. You can see calibrated ruler and the navigation overview was really popular, and I think it's quite straightforward to understand why. If you want to do a measurement in a microscope, you take a pen and you carefully make a very small dot where you want to start your measurement, and then you make another dot, and then you take a manual, like physical ruler, and you just measure. And in the computer, you can just draw a line and you have the result. So even those who didn't diagnose digitally still use that feature because it's worth opening in them digitally to just do the measurement. And then you can see various adaptation rate, what was useful. Most pathologists in the first one over there thought the image quality was quite sufficient. Of course, the ergonomics became better, which was the goal, so that's reassuring that at least that happened. But then you can see a lot more dark red here when it comes to, is the case more convenient to report or is it faster to review? And it really wasn't. So just to summarize the early work, that the pathologists there took help with their IT personnel and sort of built up this IT system by themselves. It wasn't provided by an external company. And they reached their aim with improved ergonomics, and the pathologists who had this neck problem could actually go back and work 100% again. So for them, it made a big difference. Some early problems were the main problem was the reduced speed of navigation. You couldn't really use this. If you are good with a microscope, you were usually faster with it still with a microscope. And a lot of pathologists blamed it on the computer mouse. Doesn't fit. It doesn't work, and you get tired after doing too much diagnostic work. Okay. So this led us into, we wanted to investigate this further, which led us into the second paper with the title Slide Navigation Patterns Among Pathologists with Long Experience of Digital Review. When I was searching for the related work for this paper, I found a couple of studies actually that had studied pathologists, what they do in front of the microscope before. The first quite interesting study, I think, is a study by Rebecca Crowley, who studied, I think it was 20 participant pathologists who each diagnosed a case with a microscope and had a manual video camera to show what they did. And they asked them to think out loud while they were doing their diagnostic review. And then she did a iterative coding method called protocol analysis on the verbal statements and sort of divided up it into what type of findings do they use, how do they combine what they see in order to make a hypothesis. And she did quite an extensive review of all the verbal reasoning that goes on with pathologists. So it's quite helpful to read as a first paper to understand what pathologists do. Other studies have studied eye movements. They do still images of the initial view of the slides and look how pathologists search around. And they found that expert pathologists find a few features quite quick and spend most of their time there, whereas medical students usually scan a lot more back and forth and doesn't really decide on what is important or not. And the third type of studies that exist is look at the navigation patterns, but look at the dwell. When you navigate around, where do you stop to contemplate what you're seeing and figure out that? And then you create heat maps to see what are the interesting parts. So I'm going to show you a clip now of a pathologist thinking out loud, and he's doing the third slide in a appendix case. He has already looked at 2 slides, and when you see this movie, he will go into the third and last slide. So I thought we could look at it together. Can I click here now? So that's typically how it looks like when they're reviewing. And what I see here is that I don't see a lot of dwell, I see a lot of moving around. And so the studies that have already been made sort of focused on the dwell, even though what all they do is just move around all the time. So that's why we focused our study on analyzing the movement patterns, because nobody has done that before within the domain of pathology. So we got 4 representative cases of different types. We had 5 pathologists participating. We asked them to think out loud so we would understand what they were doing. And we recorded the navigation patterns and how they zoomed around and how they pan around. And in order to help us with the analysis, we created a tool to visualize the navigation patterns, which I'm just going to show you. That one. So this is the same, if I find my mouse pointer, there. So this is the same case where we can see the zooming pattern. It's a little bad contrast now, but this usually stands out more, where it's possible for us to quickly browse through the navigation pattern, and we can step through different parts of the review and replay it, like this. So in order to understand this, we divided up these navigation patterns in small chunks, and we classified them iteratively in different types of classes. And we did it with 2 different coders to check that the coding system that we created was reproducible. And we ended up with these six classes, 3 types of panning and 3 types of zooming. And the panning one, the first one, directed panning, we call it when you go from one part to another quite quickly without looking at what's in between. So it's the sort of transport function when you want to move somewhere else in the image as fast as you can. We have cover panning when you do this systematic search of you have a feature in mind that you want to find and you want to check if it's there or not. Welcome. So if you have an area, you typically go back and forth like a lawnmower in order to cover everything. But if you have an edge feature, which only exists in the edge, you just follow the edge in a particular magnification. And the third type of panning we saw was we call it sporadic panning, when you just want to have a look around without caring too much about to cover all the area. Of course, we have zooming in and zooming out, and what we call dip zooming, when you saw something interesting and you want to zoom in on it quickly to see what it was, and then zoom out and continue whatever else you were doing. So by classifying these movements, we could then, of course, do statistics on it. This is the total time spent on each navigation type. We call them navlets, short for navigation chunk. And we see that over half of the time we're spent on these systematic search tasks. You go back and forth, and most of the time you do it in order to conclude there's nothing there, which is a quite distinct diagnostic finding that you need to incorporate in your diagnosis. And then, of course, it goes down, sporadic panning, dip zooming, in different periods of time. If we look at all the pathologists that participated, we can see at least some tendency that each pathologist have a particular style which they maintain independent of what the case is. You can see that tendency especially for dip zooming in the red, where you can see it's very consistent for the cases, whereas the edge between blue and green here, sporadic panning and cover panning, is quite fuzzy. But that also has to do with the classification scheme, where if you look at the confusion matrix, that's where you had the most disagreement between those two classes, because it was hard to separate between them. So just to recap, cover panning was most common. Much of the time you spent on concluding there was nothing there. And actually we also looked at, tried to estimate the types of movement that they did, because these were experienced pathologists that had diagnosed digitally a lot. We tried to estimate the amount of movements that they did that would have been really uncomfortable to do with a microscope. And now I don't remember the number, but it was around 50% somewhere. So if you would do it exactly the same way, it would have been almost impossible with a microscope. Which tells us something about what happens when you go from one technology to another. It takes some time in order to get these new routine movements in place in order to make that technology comfortable to use. I think the large amount of cover panning also explains why they didn't like the computer mouse in the first questionnaire. Like, if you go around and cover back and forth all the time, then you'll have to sit like this with the mouse all the time, and you get really tired. So that's why in the next study, we wanted to investigate, could we do this better? Could we do better what they had in terms of what input device they would use, and then focus on designing something that worked really well for this cover panning movement. Just to go back to the microscope, when they're panning with a microscope, they have two alternatives. And first, they have this knob here, which has an X and Y sense, so you can move the glass slide with this one. Or the more popular way is to remove this glass slide holder, and you just move around with the finger instead. Typically, young pathologists prefer the finger because it's faster, but if you get experienced and tired in your hand, you usually go back and use that one. We limit our search to input devices to devices that already existed in the market, because if we could find something that already existed and it worked, it would be much easier to make it work in practice. It's quite expensive to develop your own input devices, especially for this small target group, which is in Sweden, it's only 200 pathologists. We selected these three, the mouse as a reference. The middle one here is we call it 6 degree of freedom controller, which is basically a puck in the middle, which senses how you push it in all directions, including all the ways you can rotate it. And we had the touchpad, which of course we thought would be quite similar to using your finger on the glass slide, so it might work well. And We evaluated this in a comparative study where we had 6 pathologists diagnosing total 9 cases, fully counterbalanced. And we interviewed them afterwards, and they had to fill in the one NASA TLX questionnaire in order to, which is a way to measure the perceived workload when they were doing the diagnosis. For the NASA TLX scores, we got this result, where the mouse and the middle 6 degree of freedom controller got low scores, whereas the touchpad got quite high. For me, at least, this was quite surprising, because we sort of thought the touchpad would perform a lot better. We put in most of the development work in that one, actually. But there was something, and in the comments in the interviews we did, there was something that they weren't able to find a position where they could use the touchpad comfortably for a sustainable period. It was quite nice in the beginning, but for a sustainable period, it's quite hard to not strain your hand. If you look at the subscales, you can see the highest one is, the highest difference is the effort and frustration and performance. So you can get a feeling of so it seems true that it's the physical strain that made them dislike the touchpad. So this is sort of I think the 6 degree of freedom controller was successful because you can do the cover panning while basically having your feet on the desk and doing it really comfortably, going back and forth. So you can do that in a sustainable long period of time. The last paper, we took another approach of solving the same kind of problem. So instead of trying to make the scanning movement really comfortable to do, we tried to reduce the need of doing it in the first place. So we apply that on for particular subtasks, which is called to grade KI67 slides. And I'm going to show you a pathologist doing this. It takes about a minute just to get a feeling of what you're doing, and I'll explain what he's doing. Let's play. Okay, so he selects the slide here, which has a staining which marks brown cells, which are proliferative. It marks them brown. And you want to measure this where it grows the most, because cells are dividing. It's an exponential phenomena. Whereas the most brown is where the future growth would have been. So they do this scanning movement in medium magnifications and scan around and try to find, they call it the hotspot, where is the most proliferative cells. And I have censored the subtitle lid here because he's swearing a lot. Because this is a quite tedious and hard task. You have to keep track of what you have already seen, and you need to combine that with a movement. And he finally decides on a spot here, and he zooms in, and then you have to manually count the number of brown cells here for 200 cells. But now he's just estimating here. And then he just estimates to under 10%, which I have controlled it. It's quite accurate. So the problem, I think the main problem here is that in low magnification, you don't see the cells. So you really have to zoom in and start cover panning. So we just did a very, very simple algorithm where we separated the blue and the brown with the color deconvolution algorithm. And we made the purple paler, or the blue channel, in order for the brown to stand out. And we also put a called Gaussian filter, which has the effect of making the cells larger. So you can see here it's very similar to the original image. And there in low magnification, we have processed the image quite a lot so that the brown cells stand out. But it's really hard to get the sense of the feeling in a still image, so I'll just show you another one of these. there, and then there, and then. So if we can take the same image. This is an unprocessed image of the same kind, so you can't really see anything. So it's first when you zoom in here, you start seeing brown cells, and so you really have to work around down here. But after the processing was performed, the brown cells really stand out, and you can take the decision down here. But then Klaus told me that you shouldn't change images for doctors. They get really angry if you change their original material. So then we did so that when you zoom in here, we go back to the original image. So here you see the original image. So we provide a mechanism to verify that the processed image is actually correct. So you can follow the brown here, and when you zoom in, you can see that the brown that you saw is still there. It's quite important. I have another example here, for example. Here you can see brown dots a little bit here. You see a large brown thing there, and then another brown thing up there. And I happen to know that this one is actually an artifact. But since you have to zoom in to do the counting anyway, this is something that you will always detect. So we did a small feasibility study to check if this was okay. Is it okay to process the image and have this verification mechanism instead? And the basic result was pathologists said that the tool improved their sense of distribution. They saw something that they have never seen before, which they were quite astonished about. We discussed a lot about, is it okay to process the low magnification? And most pathologists agreed that it was, which means that you can probably reuse this design pattern for other tasks as well, where you do a processing in low magnification and then you zoom in to verify. And the third one, which was quite interesting, we had a discussion about, I asked them, how have I digitally processed the image? And then they all described the phenomena in terms of lab procedures. So the pale blue was described as something gone wrong in the lab, whereas the intensity increase of brown was, there's a lot of, you had a too sensitive immunostain here. So, which makes you question if our pathology is competent to judge your image processing algorithms, or you have to probably, because if you don't have a verification mechanism, are they then competent to detect the mistakes? Probably not. So I'm soon going to finish. I'll just say a few last thematic things first. I think about two things when I think about all these papers, and the first thing is, of course, this systematic search tasks, where I think other areas of HI could definitely learn actually from pathologists. Where, for example, I found a quite interesting study by Kaspar Hornbeck, where they studied PhD students reading and answer questions on papers, where they sort of have the same scanning behavior as we saw the pathologists do. So the systems that we come up with the mouse and so on are probably useful even for those types of tasks. For example, one of those was on the mouse. I didn't tell you earlier, but if you middle mouse click the mouse, you lock the mouse to the panning with a higher sensitivity, which makes it possible to skim through something very, very fast. And of course, you can probably, in other domains as well, it would be possible to sort of try to find a way to remove the need of doing the scanning anyway. And where one way is to sort of push the information up into a lower magnification in a smart way. In maps, they already do this. If you look at Gothenburg on Google Maps, they have a very selective algorithm in terms of what information you see on each zoom level. But then you already have semantic informations which you can query and make a decision on what should be visible. Whereas here you have unstructured image data, and you have to make up an algorithm to create the semantic structure that you need. So that's something that I think HCI can learn from pathology. But I also think the other way around is fairly true. We have other analytical domains. For example, so Klaus told me in the car here that Homeland Security funded a lot of research into studying text documents and do intelligence analytics. So there's a lot of theorizing around this. And most of it, they call it, you call it sense making, where you divide the process up in collecting information, shoeboxing it, and make hypotheses. So I actually took one of the models from one of these papers, and I took the Crowley paper and I mashed them together into this diagram, where you you have a specimen, you create glass slides, and at the end you want to create a report. And then you can do this process of finding specific images that you see that you want to incorporate into diagnostic review. And today, all these things sort of takes place in the pathologist's head. They don't keep physical track of these things. And another way of seeing this future approach would be look at Schneiderman's InfoVis taxonomy, where we could sort of say that today with a microscope, you can zoom to get details on demand, but you can't do very much more. With the digital interface, we add the overview, so you can do 1, 2, and 3, but we still have number 3, number 5, number 6, and number 7 as sort of something to really look into in the future, because this has worked for analytic professions in other domains. It's quite likely that some of it could be relevant also for pathologists now when we have access to the digital images. So at this stage, a lot of pathology labs are looking into this digitization thing. We have a lot of projects going on in Sweden, and I think the reason for that is that when we start adding up all these small improvements in order to build a better workstation, at some points you will start where diagnosing with the computer is actually more efficient than the microscope. I talked with one of our colleagues in Linköping hospital yesterday, or last week I mean, and she said that she's been diagnosing digitally so much now, and she's become really dependent on the overview. So when she diagnoses with a microscope now, she starts lifting up the glass slide a lot more to get a physical version of the overview, because she lost the ability of keep tracking where she are. And when that happens, I think this type of technology will soon be used everywhere. And I'm finished. Thank you.